Hey guys, welcome to the second part of my genetics video, and this time we're gonna do genetics. So you might be familiar with the name Mendel. He was a person who for the first time um, did concrete and organized genetic experiments using this particular species of pea, which had really distinct traits and which had a small generation type, and he observed certain things and concluded some rules of genetics. So at the moment he was unaware of what chromosomes actually were, and what he did was he used true breeding lines of bees. That is, one particular breeding line compared to another, they were the same at all the gene loci except at one particular locus. And because of that, because the others were the same, it was very much easier to get an idea of how inheritance worked. And there were like 7 traits he worked on. All of them had one dominant and one recessive trait, which made things much easier. So he was the one who laid down the dominance and recessiveness relationship, although later on there were obviously changes observed. Codominance, which didn't kind of make sense with the according to all of the data Mendel had provided, as well as overdominance and stuff like that. So he also had this thing about the law of segregation, according to which 2 particular genes, unless they were on the same chromosome and tightly linked on the same chromosome, they were likely to not be inherited together, and chances of any of the alleles being inherited being 50 to 50%. So that was the law of segregation, which we know during mitosis about independent assortment as to which of the 2 chromosomes we're going to get, that being entirely random was something he theorized. And the whole probability, how to figure out probability of how many gametes and the probability of the end trait of the, or how many of the the offspring had a particular trait. So this man, Gregor Johann Mendel, he laid the foundation of classical genetics. And how did he do that? He formulated 2 laws: the law of segregation and the law of independent assortment. And he performed a series of breeding experiments on garden pea. So I don't know how important this is, but if you can, it'd be a good idea to remember. You don't have to learn it, just that if you see it anywhere, you'd be able to remember what this was. So this was the scientific name of the pea plant that helped lay the foundation of classical genetics. Yay. Right, so he did this for 11 years in his monastery, and this garden pea was easy to cultivate, grew well in his garden, and its flowers were hermaphrodite, which means that they had both male and sex reproductive organs. Right? Must have made stuff easier. It normally self-fertilized, but it could be cross-fertilized. As the type gap between the generations was short, Mendel could raise many generations of pea plants within a short time period. Now, pea had many sharply distinct traits, which helped a lot. And each trait had 2 clear-cut alternative forms or varieties. Example, seed shape had a round or wrinkled phenotype. The plant height was either tall or it was short, and there was a particular range which made it considered as tall and a particular range which made it considered as short. The seed color could be either yellow or it could be green, and Mendel called them contrasting pair of a trait. So actually the contrasting pair of a trait is what is an allele, and he focused on 7 such pairs, right? So 7 contrasting pair of traits. So 7 genes or 7 pair of alleles. They do not necessarily have to be contrasting. They can be very similar, but for the particular plant he studied, they were very distinct. So he first established true breeding lines or varieties of each trait, right? And a true breeding variety upon self-fertilization always produces offspring that are identical to the parent. And what that means is that they are completely homozygous at each loci. True breeding lines of each trait. And in simple terms, they were homozygous at all loci, which is why on self-fertilization all of the offspring happen to be identical. Anyhow, for example, a true breeding round seed plant produced only round seeds on fertilization. Similarly, a true breeding wrinkled seed plant produced only wrinkled seeds. So on self-fertilization, all offspring identical to parents. So this is an easy enough concept, but I had this inkling that they might ask this sort of an MCQ, which is why I am repeating this again and again. All offspring identical to parent. Moving onwards. So here we have the 7 contrasting pair of traits he studied. There is plant height, there is flower color, so it can either be purple or it can be white. Flower position can be at the junction, at leaf junctions, or at the tip of branches. So that point where a couple of leaves are branching off, we can have a flower, in which case we call it axil. If it's at the end of a branch, it's terminal flower position. The pod color can be green or yellow. The shape can be inflated or constricted. Seed color can be yellow or green. Seed shape can be round or wrinkled. So basically 14 pure breeding lines of 7 characters. 14 because there were 14 alleles or 14 contrasting traits. 14 pure breeding lines of 7 characters. He cross-fertilized plants that differed in one character only. Obviously that was so he could only study that particular trait without worrying about the rest of it. right? Because he was sure that all other loci were similarly homozygous, so that wasn't a problem. And he just had to look at the one traits they differed in. And, and the offspring of such a cross were called monohybrids because only one gene loci was under consideration. He cross-fertilized a true breeding round-seeded male plant with a true breeding wrinkled-seeded female plant. And what was observed? He called it the first parental generation, and their offspring were called F1 of first filial generation. Right? And all of the F1 offspring were round like one of the parents, so that meant that a round shape of the seed was this dominant trait. And wrinkled phenotype did not appear at all because it was a recessive trait, and unfortunately you got to remember this too. Round dominated wrinkled, and the dominance was complete, and this is one of the most simplest form of inheritance, complete dominance. No intermediate offspring was found. He called the trait that appeared in the F1 as dominant and the trait that was masked in the F1 as recessive. The F1 monohybrids were allowed to self-fertilize and the F2 progeny was produced. And as a result of monohybrid cross, um, 3/4 of the F2 were round and 1/4 were wrinkled. However, the genotypic ratio was different. It was 3:1 in phenotypic ratio, but 1:2:1 for genotypic ratio. And this is kind of important. If you look over here, these are also round, these are also round. The genotype is different but same phenotype, so you can't really tell just by looking at it. And the same results and the same ratio were obtained for all the 7 pairs of contrasting traits. So then he proceeded a step further. He fertilized the F2 plants to raise the F3, right? And he noted that 1/3 of the F2 round produced only round, while 2/3 of the F2 round produced round and wrinkled in 3:1 ratio. Now this is interesting. So we know that 3/4 were round. Out of these, 1/3 of round on self-fertilization produced only round. Two by third of F2 round produced both round and wrinkled in 3 to 1 ratio. It's easy enough to see why, because we get RR, RR, capital R, capital R, capital R, small r, capital R, small r, and small r, small r. So when this is the ratio usually observed when you breed two heterozygotes. Produced round as well as wrinkled in 3 to 1 ratio. And 1 out of 1 of F2 wrinkled produced only wrinkled. Now that was obvious. And he concluded that 1/3 of F2 rounds with true breeding like P1 round, that is which was a parental generation, true breeding like P1, and 2/3 were monohybrids like F1, which is also true. Two by third monohybrids like F1, which were produced from the breeding of P1. Different P1s. So this is all very interesting and very simple, except when they actually give the question, and I for one get really confused. So some stuff you got to remember: round is dominant, wrinkled is not dominant. So Mendel's interpretations are pretty interesting, as well as the laws that he formulated. Any words on to Mendel's interpretation of his work, or more like his observations? So Mendel proposed that each contrasting form of a trait, example roundness or wrinkledness, was determined by particulate hereditary factors, and he personally called them as elemental. Now this is particularly important. Particulate hereditary factors, and we now know them as alleles, which is obvious, which I think you got already without me having to point it out. Elemental, kind of like element, but with an N at the end of it. I'm not funny. Anyhow, moving forwards, these factors carrying hereditary information were transmitted from parents to offspring through gametes, right? Obviously, each pea plant had a pair of these factors, one derived from the male parent and the other from the female parent, and both these factors together controlled the expression of a trait. He designated the dominant factor with a capital letter and the recessive factor with a small letter. Example, capital R for roundness factor and small r for wrinkledness factor. Johansen renamed them as genes. In case anyone asks you, it was Johansen, the scientist, who renamed them as what we now know them as, Johansen. And so capital R is for roundness, if they ask you is elemental an allele or a gene, go with gene. And small r is wrinkledness. Right, the true breeding around seed plant of P1 generation. P1 is the parental generation that with true breeding and the carried capital R alleles, while the true breeding wrinkled seed plant of P1 carried the small r alleles. When both the alleles of a gene pair in an organism are the same, the organism is homozygous for that gene pair. An individual with a homozygous genotype is a homozygote. So stuff we already know, nothing new. Mendel inferred that the factors of a pair separate from each other during gamete formation, so that each gamete got only one factor for each trait. So elementin was gene and factor was allele. This is also a distinction. So half the gametes got one allele and the other half carried the other allele. Fertilization was a random process. So when male gametes carrying capital R fertilized female gamete with factor small r, the complete set of the two factors for the trait was restored in the zygote. The zygote developed into F1 offspring that were heterozygous RR because the two alleles of its gene pair were different from each other. An individual with a heterozygous genotype is a heterozygote. The F1 offspring was a monohybrid for seed shape, and what that means was that it was round in phenotype but heterozygous in genotype. So this is an interesting thing to write down. The F1 generation was monohybrid for seed shape. And why wasn't it simply termed as say hetero-homo? Because it was round in phenotype but heterozygous. So we can't call it true breeding. It is a monohybrid, even though monohybrid and true breeding in this scenario have the same phenotype, but this is monohybrid because typically they are different. And so this cannot be termed as true breeding, rather we have a new term, monohybrid. These are strange terms, but I feel these are terms you need to really get into your head. An individual with its alleles also separate during gamete formation. Segregation means separation. Independent assortment means that the segregation or which one of the segregated gametes is taken by a particular gamete is completely random. So these are actually working together. The Punnett square indicates that 1/4 of the F2 progeny would have been capital R, homozygous round, 1 by 4 plus 1 by 2, 1 by 2 would be heterozygous round, and 1 by 4 would be wrinkled. So segregation is simply the law that the allele pair would separate. An assortment, random assortment, means that which of the gametes would receive which of the chromosomes is entirely random. Or one might say that the division is random. So basically really closely linked, and which is why we use these alternatively to mean the same thing in A levels, so technically they're a bit different. Now Mendel actually observed 3:1 phenotypic ratio in F2. His phenotypic data of F3 can also be explained on the basis of the 1:2:1 genotypic ratio. Mendel compared the results of all the 7 separately studied characters and found them strikingly similar to formulate the law of segregation. The two coexisting alleles for each trait in an individual segregate separate from each other at meiosis. Each gamete receives only one of the two alleles. Alleles unite again at random fertilization of gametes when the zygote is restored. Now we talked about how true breeding lines and monohybrids are different, and that when the F1, which is a monohybrid, and F1 was a true breeding and P1 was true breeding, and that F3 had both monohybrids and true breeding line. So how do we know? Because they're phenotypically the same even though they're genotypically different. How do we know which one is which? They have the exact same phenotype. What we do is a test cross. We cross the suspect individual with one that is recessive, that is homozygous recessive, and on the basis of all the offspring we decide whether it is which of the two it is. So Mendel again was responsible, and it is used to test the genotype of an individual which is showing a dominant phenotype. Individual showing a dominant phenotype is crossed with an individual showing the corresponding recessive phenotype, and the homozygous or the heterozygous nature of the genotype can then be found out. So if it's a true breeding line, all of them will show the dominant phenotype. So basically all round seed progeny, and the tested phenotypically dominant individual is also homozygous. In this way, half would be round and half would be wrinkled seed progeny. So pretty different. Half. The results would be quite distinctive. Now let's see if we're going to talk about the dihybrid cross in this one or in the next video. Let's end this over here. I'll do the dihybrid cross in the next one. Please like and subscribe if you find this to be a useful